This is the Tissues Lab, Critical Thinking, and I'm going to go over some images and some of the tissues and tell you how you should read the question and come to a logical choice amongst all the possible choices given. So here's the second one, and here is the third question, and let us go back to the first one, where it says you would expect the tissue shown here to line. Now here the key word is to line. When you see this kind of a word, it means that it has to be epithelium, because epithelium is the only tissue which lines cavities, the inside of an organ or on the external surface, for example, skin. You will be given questions. Most of the questions will say identify the type of epithelium seen here, or identify the type of connective tissue or cartilage or something like that. But occasionally you may get questions where it just says tissue, and there you have to look for clues in the question. So coming to this one, now this is epithelium, and first of course you've got to identify the epithelium to then choose which one of these is the right answer. Now whenever you're looking at epithelium, always identify the basement membrane first. And if you look at this thin line, here is the basement membrane, and this is an image which is taken from one of the sites you were asked to visit. So it's very important that you visit these sites frequently, because the only way you can make out or identify images is constant exposure to these images. So this is so therefore please look at these images very, very often. So that's the basement membrane, and these are the cells. So if you look at these cells, you can see that they are as tall as they are broad. Hence this is cuboidal epithelium. Some students often mistake this as stratified cuboidal. It is not stratified cuboidal, because they think that this is the second layer of nuclei up here. But if you look, there is no basement membrane. There's space here. The basement membrane very clearly is this one here. You see this one. These are just cells which are nuclei of cells present in the connective tissue, as are these. So that's why this is cuboidal epithelium. And the correct answer is this, because blood vessels, the lining is endothelium, which is simple squamous. The ureter is transitional, which is a stratified epithelium. The esophagus is stratified squamous non-keratinized. You must always write the correct form. If it's keratinized, you'll write keratinized. Non-keratinized, you'll write that. And large ducts carry stratified. They have stratified cuboidal lining them. And just to show you, how these other epithelia differ from the one shown here. Here I'm going to the site from where that image was taken, and here this is showing you simple squamous epithelium, where you see this is the basement membrane, and here you're looking at the nuclei of squamous cells. So you can see that these are flattened, and in case of epithelium, you invariably always find an open space, which if it's the inside of the cavity, then we call it the lumen. So this is the lumen up here. Here also, look at this, an open space, so that's the lumen. Or if it is skin, for example, on the outside, again you will see open space. So one surface of the cell is always free, like for example over here. So you can see this is a simple squamous. This shows you columnar epithelium, and again look how different it is from cuboidal. This is the basement membrane, and here the cells are taller than they are broader. So you can see a lot more cytoplasm above the nucleus. So if you were asked a question on how to give two relevant features for identifying it as cuboidal, you would say that the cells are as tall as they are broad, and the nucleus is in the center and of the cell. So just to show you that this was the image that was taken, so you can see how the nucleus is in the center of the cell. And I just want to show you stratified squamous non-keratinized. So here this is stratified squamous. The basement membrane begins here, and look at the layers of nuclei, and right up to the top you see nuclei, unlike stratified squamous keratinized where you'll see keratin. I want to show you stratified cuboidal. So here you're looking at here is stratified cuboidal. So again, look at, this was the basement membrane here. Again, you can see a lumen right here in the center, and then here are the two layers of nuclei. And just to show you transitional, and we'll bring it down back here. So this is one example of transitional. Here's another one. So notice how this is again the basement membrane, lots of layers of nuclei, and then the top layer is not flattened, as you can see here. You can see plump cells. So this is how you will see identify the different epithelia. Now let's go to the next question, and then this one says identify the tissue seen in this slide and where would you find this tissue. So the first thing here is it is not epithelium, because again you don't see a lumen, you don't see cells like you saw lining any structure, so it is not epithelium. It is also, therefore it's not epithelium. It is also not nervous tissue, because in nervous tissue you'll see multipolar cells like this which have processes, and you'll see axons, so there'll be many of this, plus the nuclei of neuroglia, so that looks very different. So it's not that. So the only possible choices you have is it is either connective tissue, any form of connective tissue, or it is muscle tissue. Now the difference between the two is you have to look at the nuclei, where they are placed, and you have to look at the cell. If you look at this image and you look at this particular nucleus along with the cell in which it's lying, you can see that it's lying in the center of the cell. Again, look at this one here and here. So all of them, so this is very typical of muscle, where a smooth muscle, where you have the spindle shaped cell and the nucleus lies in the center. If this was connective tissue, like if we know this is not cartilage, it's not a blood, so the only kind of connective tissue you could confuse this with is either dense regular or dense irregular. You would think more irregular because of all these spaces in between. But anyway, in dense connective tissue, you have these collagen bundles which lie, if it was dense regular, they would all lie in one, along the same direction. And then the fibroblast nuclei get caught in between the collagen bundles. So again, if it was like this, you get the fibroblast nuclei caught in between. So it is not dense connective tissue. Therefore, the only possible answer here is muscle, and it is smooth muscle, because skeletal muscle and cardiac muscle have striations, and in skeletal muscle the nucleus is hypolemmal. That means it is present below the sarcolemma, and plus it's multinucleated. So you can see, so one cell has many nuclei, whereas here you can see one cell is just one nucleus, and also this spindle shaped cell that you see here. And cardiac muscle is of course very different. So therefore the correct answer to this is smooth muscle, and it's in the walls of the stomach. As you know, smooth muscle is present in the walls of organs and blood vessels. So it's therefore not any of these. And again, just to show you and compare, so here this is dense regular connective tissue. So look at these things. You don't see spindle shaped cells. These are just bundles of collagen fibers where the bundles branch, and you can see a few fibroblast nuclei, not even really well seen here. And this is dense irregular connective tissue where the bundles are in all different directions, and again you see some fibroblast nuclei, very different from what you saw over there. So therefore this is not dense connective tissue. And just to show you skeletal muscle, so here is skeletal muscle. Of course this is a very magnified picture that you see over here, but you can see striations and these nuclei just below the cytoplasm, just below the sarcolemma. So that's called hypolemmal nucleus. And again, to show you another picture of smooth muscle. So here we have smooth muscle from another site that you were required to look at. So look at this. You can see these spindle-shaped cells with these nuclei. It kind of looks like fish swimming in a stream. So that's one way I look at it for you to identify. So you can see how it is very different from so this looks very similar to the image you have, but it's very different from dense connective tissue. So let's go back to question. So here it is. And let's go to the last question here, which says the function of the cell seen here is, and where would you find the cell? So obviously you have to first identify the cells and then you talk of function. Now when you get just cell, again, you're not going to get just epithelial cell or just a connective tissue, the fibroblast or things like that. Usually when you get cells, to identify a cell, it could be blood cells, or it could be from the nervous system, because identify neurons or neuroglia, or at the most probably adipocytes, because anything else will come with the whole tissue. For example, if you were to ask to identify a chondrocyte, it would be, you could see the entire cartilage. It's not present just separately without the other structures with it. So therefore, when you are asked to identify cells, usually it is blood, nervous tissue, or adipocytes. Now let us look at this one. This is very obviously that from blood, and look at these lobed nuclei, because nervous system doesn't have cells looking like this. So this is, you can see these red blood cells. These are lobed nuclei, as you can see here. So this is very typical of neutrophils, and you can see it has granular cytoplasm, but you can't tell the color of the granules. This is a lymphocyte, where you see this huge nucleus and very little cytoplasm. So this is a neutrophil, hence its function, the function of a neutrophil is phagocytosis, so that's why this is the correct answer. Now these answers where it says to conduct impulses means we were talking about neuron, which is present in the brain and spinal cord. Never just say nervous system, always give the exact location. It's not this, because neurons don't look like this. And this other choice is lay down collagen, which means fibroblast would have been the choice, and connective tissue, so we know it's not this one. Take part in allergic responses means eosinophil. It's not that, and I'll show you an image of an eosinophil and how different it is from a neutrophil. And for insulation and present in subcutaneous tissue is adipocyte. So let me show you an image of, so here's of an eosinophil. So here you can see an eosinophil, very different. It has lobed nuclei, but a nucleus is lobed, but you can see that the granules are so distinct and they're pink in color. Here's another one where you can see this. So this is again a website you are required to go to. So it's very, very important for you to look at these images frequently, so that you can then identify what the the correct choice would be. So again, also read how the answers have been written. So whatever the correct answer is, look at the choice. For example, if you were asked the function of neutrophil, you would say phagocytosis, or to take part in bacterial infections, and where is it present in blood. Okay, so I hope this review helps you.